The DNA binding protein I will be discussing today is DNA ligase. DNA ligases are an integral part of DNA replication in both eukaryotes and prokaryotes. DNA ligase's main function is to act like a glue. In DNA replication, this function is used to bind the newly generated Okazaki fragments to each other. DNA ligases have two important regions in their protein structure, the cofactor molecule docking region, which is shown here in red, and the lysine amino acid, here in purple. These specific parts of the ligase protein are what binds and interacts with the DNA to carry out its required functions. In humans, the cofactor molecule needed to interact with DNA ligase is ATP, while in most bacteria it is NAD. The lysine amino acid in DNA ligase is what interacts with the ATP bonded to the ligase to activate the protein. The slight notch in the structure of the DNA ligase, shown here on this specific ligase, is where the DNA will bind and interact with DNA ligase. DNA ligase is able to perform its function by going through several chemical reactions. For DNA ligase to bind two broken fragments of DNA, it will first need to be activated. To do this, the ligand-lysine complex shown here will need to react with our ATP molecule. Let me just label this as ATP. So we can see here our terminal amine group from our amino acid will form a nitrogen phosphorus bond with this phosphate group while cleaving a phosphorus oxygen bond, having these two phosphorus groups leave our ATP molecule. We have now formed our ligase AMP complex. AMP refers to this portion of our structure because AMP stands for adenosine monophosphate, and we only have one phosphate group left on our structure. With this new complex, we can now interact with our DNA fragments. Here we have one Okazaki fragment and a second one over here. As you can see, this complex only interacts with the 5' end of an Okazaki fragment. We form a new oxygen-phosphorus bond while breaking a nitrogen-phosphorus bond that we just made in the last step. This generates the DNA adenylate complex. DNA ligase then catalyzes the final step of this function, which is the formation of a new bond between the 3' OH and the 5' phosphorus. So as you can see, our OH group is forming a bond here with the phosphorus, and then we're cleaving a phosphorus oxygen bond here, thus removing our AMP molecule fully from our DNA. This ends up producing a new phosphodiester bond between two fragments, gluing them into one continuous strand.